I'd like to introduce my colleague, Lee Gettler, who's an assistant research professor in the Department of Anthropology, who's also part of the planning committee. He was a student of mine when he was an undergraduate. He's a delightful fellow. You'll have to get to know him with his new baby, too. So here's Lee Gettler to introduce our next speaker.
Good morning, everyone. It's my great pleasure to introduce Dr. Elizabeth Ann Quinn. Dr. Quinn is coming to us from the Department of Anthropology at Washington University in St. Louis, where she is an assistant professor and the director of the Biomarkers and Milk Laboratory. Along with her exceptional record of scholarly, scientific, peer-reviewed literature production, she also writes an excellent blog where she translates lactational science for both her colleagues like me who are not an expert in said science, as well as for the public. So I can see EA has her Twitter handle up here, so please look her up on Twitter and look up her scholarly production and her blog. It's a real treat to have Dr. Quinn here with us. She really is a pioneer in terms of thinking about milk not only as the kind of amazingly complex and incredibly important nutritional component that I think all of us agree that it is in terms of being elegantly designed to help our human infants grow and develop, but also thinking of it as a signal and thinking about how mother's milk is calibrated based on her own developmental experiences, the cultural context in which she lives, the ecological circumstances in which she finds herself, and the way that human variation, that between mother variation may be specifically calibrated to help optimize infant development for those local ecologies. So EA, Dr. Quinn and I went to graduate school together, so I'm not accustomed to referring to as Dr. Quinn, so forgive me for the EA reference there. Dr. Quinn brings not only evolutionary theory and kind of social cultural approaches to her work, but also is working within the concepts and theories of developmental plasticity and the developmental origins of health and disease. So the integration of all of these approaches and thinking about milk as a signal that that can allow short-term adaptation over multiple generations that kind of fills in the gaps between homeostatic regulation and the type of evolutionary change that we think of emerging via natural selection is really kind of a pioneering approach. EA was a few years ahead of me in graduate school at Northwestern University, and I learned a great deal from her about being a professional scholar, being a teacher, being a lab manager and technician. So it's so great and such a pleasure for me to welcome her to Notre Dame. Thank you.
Good morning. Can everybody hear me? I'd like to start by thanking the organizers for this opportunity to come and talk about my work. And I should probably start with the caveat that now for something completely different. We've spent most of the last 2 days discussing really child psychological development and really more aspects of the mind and brain and those kinds of functions, and I don't work on that. My research is primarily on breastfeeding, and more specifically, I'm really interested in questions of childhood obesity and related health, particularly development during infancy and the risk factors that may pose for not only childhood overweight, but also type 2 diabetes, metabolic syndrome, and these we know are very important in terms of thinking about child flourishing. Child flourishing, really, obesity is going to be a huge challenge for that in the United States. At present, about 18% of US kids are estimated to be overweight or obese, and these children are at increased risk for numerous cancers, for type 2 diabetes, and are actually projected to have shorter lifespans than their parents, something that is essentially unheard of in terms of the history of public health. And so one of the questions turn on my clicker. One of the questions I kind of come at this from is, well, what can we learn from lactation about what are kids supposed to be eating, about hormonal signals, and really thinking about how does early life influence our development over the lifespan. And this really started out as developmental plasticity or developmental programming. It had its origins in the fetal origins hypothesis, or the Barker hypothesis, as you may have heard of this, really illustrated quite well by this Time magazine article in 2010, which essentially said how the first 9 months of life shape the rest of your life. And this was purely a fetal model for much of its original history. And the idea was that nutritional insults during gestation, growth faltering, any kind of disruption of nutrition, including stress, was going to increase the offspring's risk of low birth weight and subsequently of cardiovascular disease, diabetes, obesity, metabolic syndrome, any number of factors. Well, now we know actually that it's probably more something like this. And those of you familiar with some of the more contemporary public health models know there's the big Gates Foundation push for the first 1,000 days. And that is the observation that the first 1,000 days from conception are incredibly important in terms of long-term childhood health and nutrition. So really when we're starting to talk about a lot of this with breastfeeding, not only in terms of the neurological development that we've been discussing the last 2 days, but really it's important to think about the physiological development and growth happening over this interval. And so when we begin to be aware of how important the postnatal period was, that everything just didn't stop at birth, this was kind of posed as one of two questions. And this question was pretty straightforward. Are the influences that we see on long-term development during the first year plus after birth being driven by nutrition or growth? Am I pinging loudly? Am I really hard to hear? Okay, better? Okay, perfect. The question is, well, is this being driven by nutrition or growth? And one of these models was the growth acceleration hypothesis. And what this proposed was that when we see physiological changes associated with prenatal nutritional deprivation, that is low birth weight or some other form of early growth faltering, catch-up growth and acceleration of growth after birth, and this is weight gain, length gain, was associated with problematic outcomes. These kids were more likely to develop cardiovascular disease, obesity, and the mechanism was thought to be some sort of insulin resistance. Alternatively, there's an entire set of scholarship coming out suggesting that, well, maybe it actually has more to do with the composition of the postnatal diet. So exposure to a high-fat diet or a high-protein diet is actually going to alter metabolic function and how our bodies regulate fat, carbohydrate metabolism, and even deposition of body fat. And really, these two questions are complementary because for much of infancy, growth and nutrition are intrinsically linked. When we tend to think about early nutrition, we think of these kind of dichotomous models of breastfeeding versus formula feeding. But formula is in fact a fairly recent development. And we know there are some well-established differences in function during infancy and even in childhood growth between breastfed and formula-fed infants. Breastfed infants tend to have faster, that is greater weight gain in the immediate postnatal period, and then growth slows down towards the end of infancy, whereas formula-fed infants have an acceleration towards the end of infancy and often do not have that postpartum weight loss, that immediate postnatal weight loss that we actually think is important in some of this programming. So what is it then? The original models for this was that it could be entirely linked to the nutrients and the energy available in infant formula compared to breast milk. Now, I know most of you don't spend as much time as I do thinking about the awesomeness that is human milk or in the amount of sheer science that goes into infant formula. These are kind of things we take for granted. But there are some key differences between them. Number one, breastfeeding is a living substance, and we'll go into this in more detail in a moment. But number two, there's actually more protein in infant formula. And so part of this early hypothesis was that infants who were receiving commercial formula were receiving higher amounts of protein. And so this was accelerating their growth. And this acceleration of growth wasn't uniform. It wasn't all tissues growing at the exact same rate and ending up at the same size, just a larger infant. It was instead very specific tissue types growing at a faster rate, particularly an increase in weight gain and fat mass. So what is it then about milk if maybe it isn't nutrition? Kind of already foreshadowed this, and Dr. Gettler did as well, in terms of saying, well, I'm really interested in these questions of milk as a signal. So maybe there's something else going on between just the nutrients in milk. And that is not to discount the nutrients in milk as being important in this conversation, but there are many things in milk besides just the kind of macronutrients. And so hopefully you can see this is a lovely separated sample of human milk. You've got protein concentrated on the bottom, lactose and water in the middle, and then a fat layer. And human milk contains about 4 to 5% milk fat. The kind of reference is about 4.2 grams per 100 ml. About 7.2 grams of sugar, and about 1.2 grams of protein per 100 ml. But that's not all that's in milk. Milk is predominantly water, and it is also chock full of micronutrients, of fatty acids, of oligosaccharides, of probiotics. There's bacteria in milk. There are immune factors, such as secretory IgA that we know support the infant's immune system. There are monocytes in milk that we know specifically target pathogenic bacteria. There are sugars in milk that we can't digest, but are in there in order to feed our gut microbes. There are even, hold on your socks, stem cells in human milk. Pluripotent stem cells in human milk. So milk is this incredibly complex medium that has this tremendous potential to convey considerable information across generations. And when some of the early developmental programming models were coming out, we were thinking about, well, what an excellent conduit the placenta was. This internal gestation in terms of communicating signals across generations. Well, milk is an extension of that because of the sheer number of factors in it that can communicate a signal. And so we know, leaving aside signaling for a moment, that there are tremendous health benefits for human milk. These are the ones you're probably most familiar with. These are short-term benefits. We know globally, not just in highly pathogenic environments, but also in the United States, which is a pretty resource-rich, low-pathogen environment, that breastfed infants, or infants receiving human milk, because there now really is a distinction, have reduced rates of diarrhea, respiratory infections, ear infections, and all-cause mortality. Now, this is especially pronounced in highly pathogenic environments, but it's probably is explaining the lack of human milk is probably explaining about 1,000 excess deaths in the United States. And that's even discounting some of the data suggesting the interactions between human milk and other factors such as sudden infant death syndrome and even later childhood cancers. So that's kind of the short-term benefits. In terms of the long-term questions that Dr. Gettler was setting up in the intro, well, what then can lactation contribute to this kind of larger question of obesity, of type 2 diabetes, of all of these metabolic conditions that are on the rise in the United States? And we know from some very elegant studies that breastfeeding is associated with a reduced risk of type 2 diabetes, of stroke, of cardiovascular disease, of insulin resistance, and of other characteristics of the metabolic syndrome. We have mixed evidence for whether or not breastfeeding is truly protective against obesity, and I do want to kind of talk to that point because probably most of you saw in February the Social Science and Medicine paper that came out and said breast isn't best. It was all over Twitter, it was all over the news, CNN ran a special story, and what they had done was they had analyzed some data, a sibling cohort model, and found that there was no protection of being breastfed on risk of obesity. And this was presented as a definitive answer of, oh, well, that intervention doesn't work. In fact, it's part of this ongoing conversation we've had where we have really mixed evidence for, is breastfeeding protective against obesity? And really, it's not that we have mixed evidence in terms of outcome, it's that we have inconsistent definitions of what it means to be breastfed across studies. So we have known from the work of Fall et al. In global populations that in populations from developing countries and developing economies, there's no protective effect. Meanwhile, a lot of work in the United States, including an alternative sibling difference model, has found a protective effect of breastfeeding on obesity. And the most likely explanation is that what we're actually seeing are difficulties in classification. In many studies, being breastfed is, did you ever breastfeed your child? That means if you let them nurse once, you're in the breastfeeding group. And the entire rest of their exposure was infant formula, you're in the breastfeeding group. If you nurse for 5 days, you're classified with someone that nursed for 500 days. And that's a terrible, terrible way to quantify an exposure. But the overwhelming majority of studies simply treat breastfeeding as a dichotomous variable and often even don't quantify if there's mixed feeding going on, the duration of mixed feeding, the timing of supplementation. So the fact that we're kind of detecting any signal at all probably suggests that there's something important and interesting going on there. So what does this mean then for some of our larger questions? And what I want to do today is actually walk you through some of the evidence for the short and long-term benefits of this kind of lactational programming and what we think are some of the mechanisms going on that may influence these protective effects of breastfeeding against obesity. I will ask that I am presenting some of my students' work. Please don't photograph some of the graphs because it's still in review and I don't want the cat to get out of the bag before the cat is out of the bag, simply out of respect for their work. So what's in human milk? As you can see from this easy-to-read slide, a whole heck of a lot. And that's really the best way I can summarize this is to show you this slide that you can't read because it's got too much writing on it and tell you that it's actually missing about 500 things. My lab is 30 feet in length, and we have the definitive list of everything found in human milk running the length of a wall. And eventually we're going to have to commandeer a second wall, just because every year we find something new. 2 years ago, we didn't know there were stem cells. Now we think that they may actually be able to hone to specific organs and reshape organ development. So when we're talking about what's in milk and the potential for signals, we're talking about multiple levels of signals that can convey multiple types of information, and that this information may be incredibly important because of the kinds of signals and the information it's providing. So the kind of first argument for what are all these hormones doing in human milk and how are they contributing to reductions in the risk of overweight and obesity, type 2 diabetes, to improved insulin resistance is that they are involved in appetite regulation. And that is, we know when infants are feeding at the breast, they do not take all of the available milk. It actually looks like, based on some fantastic controlled studies where they got women to generously hook their breasts up to ultrasound devices, to displacement studies, even the new volumetric measuring equipment that's coming out of Israel now, they were actually able to determine that the infants were only taking about 70% of the available milk. So infants are regulating what they're taking in. It's not dependent on what's available. This also makes us feel good because when we go and ask mothers for milk samples, we know we're not literally taking food from a baby's mouth. There's a 30% window we can borrow from. Second, many of the hormones that ultimately we find in milk are metabolic hormones you've probably heard of: leptin, adiponectin, insulin, probably best known for its role in diabetes. And these appear to regulate appetite, not only at the level of the stomach in terms of gastric emptying, but in the ways in which they're binding to receptors at the blood-brain barrier and regulating appetite, as well as I said, gastric emptying, and even interactions between different hormones. So this was one of the kind of big models for a while. And the other thing we know is that the concentrations of these hormones in milk, for some of them actually changes over the course of a single feeding. So you start to get either an increase in some and a dilution in others that may provide important information to the infant about, well, how full are you? How much have you taken in? And may be incredibly important in allowing for this regulation. But more than that, they may also be a signal of environmental resources, of maternal resources, and literally interacting with infant physiology. Particularly coming out of animal models, we know several key metabolic hormones found in milk can actually survive digestion. They're either packaged into the milk fat globule or often found with a sugar coating to protect them from digestion, as well as changes to the pH of infant stomachs protect these hormones. In animal models, we detect them in circulation. So you can give a mama rat radio-labeled metabolic hormones and detect them in circulation of the pups. So not only are they surviving, but they're entering infant circulation and possibly targeting different tissue types. And while this may have been best worked out for immune factors, they're only one part of the story. And we know that immune cells in maternal milk can actually train the immune system to target specific antigens. And there's some very elegant work coming out of the Hind group, specifically looking at species exposure to certain pathogens and species-specific secretory IgA in milk and then infant production. So when we are looking at these prime candidates, and for simplicity's sake, I'm only going to focus on 3 today, and really only 2 of these, there are hundreds of potential candidates. Every year we learn a little bit more, and every year we work this narrative out a little bit more. But frankly, some of these were just discovered. We have maintained for a very long time that there is no insulin in human milk because insulin is a very large molecule, and it cannot get into milk by passive diffusion. So it's having to be actively pumped into milk. So it was assumed that it wasn't there until we actually tested for it and found that it was there, which then leads to the subsequent observation that it must be actively put into milk. The other two I want to talk about today are leptin and adiponectin. Both of these are mid-sized molecules, but they appear to be regulated within the mammary gland itself and have some important functions. The reason we think these have important functions is that we actually know from observational work in humans, and as we all know, you cannot ethically randomize human infants to breastfeeding formula. You know, we can't. In an ideal world, we would be able to better model this, but it's unethical. So a lot of times what we're doing is looking at observational and correlational studies. And in this particular instance with these hormones, what we've generally been able to do is look at the presence of the hormone in milk and child-based outcomes, both at a single point in time and also longitudinally. So I'll show you a mixture of the two data as well. So as I said previously, insulin is the least well understood of all of these molecules. There's about 6 papers out that have looked at insulin in human milk, and 3 of them were arguing that we need to put it in formula. So we're talking a very limited pool of data. Perhaps the definitive source on this is that we know it is not coming from the mammary gland itself. It's being recruited from circulation. So there is not mammary-specific insulin synthesis because mothers with type 1 diabetes only have synthetic insulin in their milk. And if you ask those women to generously not take their insulin for a couple of days so you can measure the insulin in their milk, it disappears. There's no insulin in their milk. Now, in non-diabetic mothers with normal insulin control, what Fields and Dimerath have shown is that milk insulin is inversely associated with BMI and infant weight, but also inversely associated with lean tissue mass. So it's not having a uniform effect on all tissues. It's instead specifically reducing, or it's instead specifically correlated with lean tissue mass, but not fat mass. So as insulin increases, lean tissue mass is decreasing. And this is quite new data. Most of the data we've had up to this point has been purely based on weight or BMI. Thanks to Field and Dimerath's DEXA studies, we're actually starting to be able to differentiate tissue type, which is probably going to be incredibly important in starting to think about what are the effects of hormones on growth, and that all growth isn't equal. That with these, when we talk about an effect on weight gain or effect on BMI, these are broad brush strokes of very finite tissue-specific processes. One other potential consequence for insulin in milk is that it may be involved in appetite regulation because infants can actually thermoregulate through feeding. And in conditions where infants may be exposed to unusual environmental conditions, we may see some variation in insulin levels in milk to promote increased feeding for the thermal effects of feeding. At least that's one of the hypotheses. So moving on to our better known aspects of human milk, the next one that's been really well described is leptin. And remember how I said it's unethical to randomize kids to formula or breast milk? This study did that. In the 80s, these were all preterm infants in the NICU, preterm low birth weight, and they were randomized to just formula or banked human milk. Now, 75% of them, 75% of the formula-only group was also receiving human milk, but they were not allowed to receive milk from their mothers. And it was a randomized trial testing regular formula, preterm formula, and banked human milk. So we're going to throw out the preterm formula data because it was a horrible experiment. It failed miserably, and they probably made a lot of kids sick. But if we look at the no formula and the formula, so this is standard formula, no formula, and these aren't data from infancy. These are data from when these kids are 13 years old. So we've jumped ahead, well, 13 to 16 years into the future based on how they were fed as infants, what's going on with them physiologically. And the kids that receive no formula have lower leptin levels. They're also producing less leptin per unit fat mass, which we think is protective against insulin resistance. And perhaps most interestingly, they demonstrated a dose response between intake of human milk to the liter and leptin synthesis 13 to 16 years later. So the graph here these are tertiles of milk intake. Were you in the lowest, the middle, or the highest? And the amount of leptin per unit fat mass being produced 13 to 16 years later. So pretty interesting results. It's at least persisting until puberty. Well, what happens next? And these are some of our work coming out of the Cebu project that you'll hear about from Lee Gettler as well. As he said, we went to graduate school together. We also worked at the same field site in the Philippines. So these are data on 1,380 21 to 22-year-old Filipino adults that have been followed for their entire lives. So they were enrolled in the study as third trimester fetuses, and we think about 57% of them are still in the study, Lee? Yeah, about 57% of these individuals enrolled as fetuses are still participating in the study. And they were followed extensively over the first 2 years of life. Every 2 months, they were visited, nutritional data was collected, anthropometric data, so we have a really good idea of how well they were growing and what they were eating. So what we've done here is we measured their plasma leptin at age 21 to 22, and we broke the amount of fat mass they had into 4 quartiles. We then stratified them based on how long they were breastfed for as an infant, with the orange being they were not breastfed, and the dark gray being they were breastfed for greater than 18 months. And the study was truncated at 24 months, so they may have been breastfed to 3 or 4 years. We have no data to tell us past 740 days of life. So if you are a very lean individual, there's no effect. It is only when we get to this higher quartile of fat mass that we actually start to see that the leptin per fat mass, the plasma leptin levels in the highest fat mass are much higher in those males, sorry, I forgot to mention that this is males only, who received human milk for 18 months, compared to those who received formula. So much higher if you receive formula than if you received milk. 22 years later. Now, here's the kicker to these data. These are incredibly lean individuals. Mean BMI was 18.7. So most US individuals are over here. This would be the lowest tertile in the United States. So right where we're starting to see this threshold effect in Cebu is what we would consider the mean for the US. And here are the data for females. Not as dramatic an effect. And the Xinghal data I just showed you previously does not have sex-specific. They combine sexes, which may have also limited their analyses because what happens between 13 and 16? Puberty, right? So all sorts of issues there. But looking at adults, and that's why we did the sex-specific analyses, there is no real association here in females. So keep that in mind because it actually is going to come up in just a moment. So what do we think is going on here? Well, probably part of what's contributing to this are hormonal differences experienced during infancy in breastfed versus formula-fed infants. And so we know that if an infant is breastfed, they tend to have lower total energy expenditure, but this normalizes by 12 months, so it's probably related to the cost of digestion. They tend to have slower growth, but they have more lean tissue mass than fat mass. They also have lower circulating levels of a lot of hormones involved in growth regulation and insulin sensitivity. So, if breastfeeding is protective, and we know there are hormonal differences, is there the potential then that the hormones in human milk are actually what's leading to those rather large differences we're seeing, at least in males? And the best evidence we have right now is that a lot of this is being driven by hormones. In 5 years, that statement may be completely wrong, but based on the data we currently have, we think these pathways are hormonally driven. And we've got some very elegant animal models that allow us to look at this. So what Pico and Pello did was they randomized rats. These are actually out of order. So they randomized rats to either oral leptin within normal physiological doses given to rat pups in their water or control saline. These rats were then randomized onto a normal diet or an obesogenic diet. So if you received leptin and then went to a healthy diet, you become a lean rat. And this is an obesogenic rat phenotype, or obesogenic rat genome line. They're prone towards obesity. If you got a saline control and you were on a healthy diet, you had an intermittent body composition. But if you'd received the leptin and you were on an obesogenic diet, perhaps most importantly, you ended up with an intermediate body composition. However, if you were on control and on an obesogenic diet, you developed obesity if you were a rat. And so we'll jump back. These are the actual numbers in terms of the amount of adipose tissue these rats had by group. So this was the normal feeding leptin group and the high-fat control group. So the obese rat to the far left of the screen, or your right, yeah, your right of the screen. So from this oral dosage study, and the original work was done injecting rats with leptin, and now we've skipped ahead to, just giving it to them in the water. It suggests then that what's going in during these critical periods may be important in terms of that long-term programming. So I don't have a time machine. I can't go back in time and ask this, but what we can do is actually start to put together some of this data to think about, well, what's going on with early hormone exposure, growth during that period, and later growth. And this is the work of Mireles, and this is on a group of 28 Spanish infants showing the association between milk leptin levels and BMI. And as mother's milk leptin increases, BMI decreases. And here, so how does this make sense? And I'll show you ours in just a moment. But I want to provide a little bit of a caveat here. And this was one of the first models we had. This is how we started thinking about these processes. And the data we have, that weird acronym that we discussed yesterday, our data are weird. Our data, just for leptin, that's the best worked out story, comes from about 17 papers, and about 3 of those are non-Western populations. And I've circled 2 of them here to show you that there's variation across populations in this association between maternal BMI and milk leptin. So maternal BMI predicts milk leptin. I reversed the order of 2 slides. And then milk leptin produces infant BMI. However, increased maternal BMI predicts higher milk leptin. Higher milk leptin predicts lower infant growth. And we think there's a sex-specific effect. And this is some work we've got coming out this month in the Journal of Human Lactation. Females are shown in orange and males in gray. And what is evident here is that as milk leptin increases, infant weight gain is infant weight is decreasing only in females. And this had not been previously described. We think because most studies have between 20 and 35 infants, and we had 116. So we were actually able to look at these associations with a little bit more clarity than prior studies have been able to do. So perhaps sex-specific programming going on. And we know from the work of Trevino-Graza in 2010 that there's a typical leptin fall in infants. So around 3 months, leptin levels in infants drop, unless you're a breastfeeding female infant. So if you're a breastfeeding female, or if you're a formula feeding female infant, your leptin level falls. If you're a breastfeeding male, your levels fall. But not if you're a breastfeeding female infant, which is the only group we continue to see this association in. So there's kind of a lot going here in terms of this narrative of leptin that we don't have worked out, largely, I would argue, because our data have been limited in terms of population variation. And these are showing you the trends in the same population. So the association between percent body fat and milk leptin, and then that same graph for mothers. So kind of the next thing we need to do here is figure out if your early nutrition influences your leptin levels in your milk, or your circulating leptin levels, or influence this relationship and become part of this longer-term nutritional signal. Finally, as I said, there's multiple aspects to this narrative. One of the other things we know is going on is with milk adiponectin, and adiponectin is also produced by fat cells, much like leptin. These are data from Wu et al. Showing the association between BMI and milk adiponectin in two populations, the US and Mexico. And here again, the argument has been that maternal body composition is positively associated with milk adiponectin, but not consistently. And here again is data from the Philippines. Could have shown you my data from Tibet as well. And in these very lean populations, we simply don't see an association between maternal BMI and milk adiponectin. And I know this scale looks huge. This scale is in nanograms, and this is in picograms, so just divide by 1,000. So when we start to look then at what is adiponectin doing in terms of infant weight for age, here's Wu's data again, showing that milk adiponectin predicts infant weight for age z-score with different patterns for individuals in Mexico and individuals in Cincinnati. And we see this also in our very lean individuals from Cebu. And you'll notice that all of these kids are losing weight. The US kids have positive weight for age z-scores or very small negatives. The kids from Mexico have slightly more negative. And then when we jump to Cebu, we've got 20% of our sample that's wasted. So these are kids with marginal nutritional status. We're still seeing an association between adiponectin and infant growth, but we're not seeing the inverse association characteristic here. We're instead seeing a positive association. So at very low levels, milk adiponectin is protective against growth. At very high levels, it's inversely associated with growth. And probably we think a very similar pattern is going on with leptin, is that these associations are important because across the distribution, these metabolic hormones may be important not only for influencing tissue-specific types of development, but also for their effects on immune function and even on protection for environmental factors such as seasonality, where it may be highly adaptive to have more plasticity. So what does this mean in terms of the US models we're talking about? Well, kind of the take-home message is, if we start to think about this in the larger context of infant formula, how is infant formula comparing to what's known about breast milk? And what infant formula is essentially reading as is a blank. We know there is no adiponectin in infant formula. We know there is no leptin in infant formula. We know there is no insulin in infant formula. People have published those data. I can do it in my lab. There's nothing. So the infant is not getting these signals. What they're getting instead is a blank. So what does a blank mean? And what we think a blank means, based on the associations between maternal BMI and these milk hormones, is a blank means a very lean mother, a very marginally nourished mother, and probably a very marginal environment. And so that's going to have effects on child growth and on child development, and may even promote very different types of growth and very different tissue prioritization. If you're in a miserable environment, it may be incredibly adaptive to gain a lot of fat while you're dependent on another individual's metabolism to buffer you from that environment. And then you can burn it off during weaning. You've got it for seasonality. I work with high altitude adapted Tibetans where there's marginal nutritional status most of the year, there's hypoxia. Those kids are prioritizing fat, and those mothers are missignaling to those infants. Their body fat and the leptin and adiponectin in their milk don't correlate. And we know from some excellent data coming out of the Gambia that we see the exact same things when mothers start losing massive amounts of weight in terms of nutritional seasonality, is they start dumping adiponectin in their milk to slow down child growth. So that mothers and infants are actually interacting and in some ways manipulating one another's signals. But bringing this back to this question of obesity, a blank then in infant formula is a signal that the nutritional environment can't support you or is very marginal. It's going to promote these specific tissue types that are actually completely mismatched to our current environment, where it's like, you know, here, have a Twinkie. I know, Twinkies went out of business, but have this incredibly rich nutritional status, so we are those missignaled mice. And part of the importance of understanding this and putting together all this breast milk science and doing more comparative work is to figure out how to protect not only breastfeeding infants, but infants whose mothers can't breastfeed, either because of individual choice, because of social factors, because of economic factors, but taking this data and improving nutrition for all kids. That's it. Thank you.
I have a question. First of all, I loved your talk, but I'm very biased because I'm very interested in breastfeeding also. So most of what you're talking about is sort of the hormonal landscape and how that's driving the process. Has there been any work, or have you done any work that's looked at some of the behavioral variation in terms of not just how long one breastfeeds, but we know that in populations, and I know you know this too, in different populations that there's big differences in terms of the frequency during the day and over the 24-hour period of breastfeeding, just, you know, bouts of breastfeeding, how many. Is any of that meaningful in this picture, or is that just sort of written off to cultural differences that don't? I mean, I sort of imagine that there must be some interaction between culture and biology here, but do you know of what the implications? We have no idea. That's what I thought. I mean, that's what I was guessing.
We need to go there and figure it out.
Yeah.
Part of this, though, is it's also incredibly difficult to study lactation, and it's incredibly invasive. So once upon a time, they would go into the field and they would put mothers in a metabolic unit and take every time and tell them every 4 hours pump, and we're going to take away that milk, and they'd give the kid formula. And now we have ethical nightmares about that.
Right. But it's not very invasive to observe behavior, though, so you can actually get a pretty good, not necessarily at night, a good count with populations that aren't going to keep track of it for you, but during the day or during a 12-hour period, you can catalog it pretty well. You don't know how much is coming out, but you can time all the...
And we've got some pilot data on 8 very willing moms who let us follow them around for a couple of days, different chunks of days, and gave us samples every time they nursed, and let us weigh the kid and observe them. And we're just starting to kind of put all that together and figure out what's going on with that in terms of regulation. So the answer is we don't know.
But you will. But it sounds like you will soon, so that's exciting.
On 6 individuals from a weird population,
okay.
Yes.
Thank you.
First of all, I want to thank you because I was bottle fed and now I know why I like to eat so much. So appreciate that info. My question is, I've done some very minimal research on my own on dairy, cow's milk versus other kinds of milk. Have you been able in your research to find differences in soy-based formula, dairy-based formula, et cetera?
So we actually don't work with any formulas.
Okay.
We're only working with breastfeeding moms, really from around the world. So in terms of the effects it has on child growth, I believe there is some literature on soy versus dairy, but I do not know it enough to speak to it.
In your information today, though, when you're talking about formula and the content of the formula, the blanks, so-called blanks and whatever, do you know, was that dairy or soy?
The assumption that there is no signal has been based on work in dairy.
Okay.
It would be reasonable to extrapolate that
Yeah.
to soy. However, with soy, you may have the added issue of the phytoestrogens, which may read as sex steroids. And certainly, I didn't even get to touch on it today, but Amanda Thompson has some brilliant work on hormonal regulation and development during infancy, including sex steroids.
Amanda Thompson?
Yes.
Thank you.
Hello, I'm Henrik Norhol from Copenhagen, Denmark. Question of colic and milk, and also the frequency of feeding. Niels Bergman has written an article where he argues that the newborn stomach volume is about the size of a walnut. And the old idea that you should sort of have a 3-hour, 4-hour spacing in between feeds. But basically, the child would be very hungry and overeat, and that would affect the whole digestive systems. And it's interesting that colic apparently peaks in the evening and around 6 weeks. Right. Anything about distance or the spacing between feedings and how that may affect how the child absorbs the nutrients and uses it?
That also has not been quantified. One of the kind of thoughts about this is that the scheduled feeding, which is the kind of every 4 hours, may be disruptive to appetite regulation, either because you have gastric stretching because the infant's taking in so much, or because of what it's actually signaling to the mammary gland. The accumulation of milk, for some women, not all, some women can certainly go 4 hours, others can't. They start to accumulate a factor in milk that actually downregulates milk production, and also the accumulation of milk in the cells alters how the cells bind to prolactin, so they actually decrease production of some of these hormones if they're actually producing them in the cells. Like insulin, they're not. So we don't know in terms of spacing, if spacings of breastfeedings, if that's influencing the hormones in them or not.
We'll have to stop now. Thank you again.